Okay, welcome back. In this lecture, we're going to start our discussion of the complement pathway. Complement, as I've said, is a really evolutionarily ancient form of host defense. It involves quite a number of different proteins, all of which conspire to uh, mount several effector functions of the innate immune system, things like phagocytosis, and also a cool process called the membrane attack complex. But there's a lot going on with complement, so what I want to do in this lecture is start by talking about the major themes, the way the pathways are organized, and we'll start by focusing on one of the pathways that is known as the classical complement pathway. So what does complement involve? So your book identifies three major stages of complement action, and I think this is very helpful. The first is we need to get a complement reaction started. So, as with most immune responses, we need to start by the recognition of non-self, so some kind of pattern recognition trigger. And as it turns out, bacteria and other pathogens have things on their surface which are very attractive for the complement pathway to begin attaching to. As we'll see, antibodies can also be a trigger for complement attachment to cells. But ultimately, once complement gets started, and there are many different ways that it can, and we're going to go through each of those, but once a complement reaction starts, all of the various ways converge on a single midpoint, and that midpoint is the formation of an enzyme that we call the C3 convertase. And so the C3 convertase is part of an overall protease cascade. And so what we'll see is that many of the complement proteins that we'll learn each have proteolytic activity. So proteases are enzymes that cleave other proteins. And so many molecules in the complement cascade, their function is to cleave the next molecule in the cascade. So that'll be a common theme. But again, we're trying to stay big picture here. When we form a C3 convertase, based on its name, is an enzyme that's going to convert C3. When we convert C3 into its products, those products of C3 are going to induce the three major effector mechanisms of complement, which are the induction of inflammation or the recruitment of immune cells to the infected tissue, phagocytosis through the process of opsonization, as well as membrane attack. And so complement can also basically poke holes in the membranes of cells and cause them to die. And this is a very effective way of killing bacteria in particular. So again, big picture here, we start with a pattern recognition trigger. All of these different triggers, they have different molecular mechanisms, but they all result in the formation of a C3 convertase. And the C3 convertase enzyme, through various molecular mechanisms itself, induces three major effector mechanisms: inflammation, phagocytosis, and membrane attack. So let's start by talking about how does the complement pathway get started. Well, as it turns out, there are three different complement pathways, three different initiation sequences, basically. They include the lectin pathway, the classical pathway, and the so-called alternative pathway. In this lecture, we're going to focus on the classical pathway, which was the first to be discovered, although somewhat ironically, it's the newest in evolution, but it is called the classical pathway based on its discovery. It's also sort of the most straightforward one, I think. So it's a good one to start with, just to identify the basic principles at work. But we'll get into all these details in a little bit later. But ultimately, whether we start with lectin, classical, or alternative, they all converge, as I've said, on the formation of a C3 convertase. And the function of a C3 convertase is to take a molecule called C3 and to proteolytically cleave it into two subunits, C3a and C3b. In these two subunits, C3a and C3b, will then go on to initiate the signaling that results in the effector mechanisms of complement. All complement pathways converge on cleavage of C3, as I have here, and C3a and C3b, the products of that cleavage, go on to either promote inflammation in the tissue, phagocytosis through opsonization, or the formation of this neat structure called the membrane attack complex. So we'll have a whole lecture dedicated to the effector mechanisms of complement. But in this lecture, we want to start more at the beginning. How does the complement pathway get started? And as I said, we're going to focus first on the classical complement pathway. So the classical complement pathway starts with a molecule called C1, and this is very easy to remember. For the most part, the numbers of the C's in the classical pathway go in order, with one exception, as we'll see. But it should be easy to remember that the classical complement pathway starts with C1, complement 1, basically. Um, Complement 1 is what actually binds to the cell initially, either directly to the cell or usually binding to an antibody. So if we look at the various subdomains of C1, we see there's C1q, which is kind of the stalk domain on this kind of upside down bouquet shape, or maybe if you prefer like a blown out umbrella. Either way, these molecules really do look like this, so we can see them under the electron microscope. This is where we how we know they have this neat shape. But the kind of stalk region, the C1q region, is the region that actually binds to antibodies. In contrast, C1r and C1s make up the proteolytic domain of the C1 molecule. So they have the protease activity, and they actually cleave the next protein in the signaling cascade. So we can look at it this way with my drawing, which isn't quite as pretty, but ultimately C1 is binding to antibodies on the cell surface. Remember, that is happening through C1q. C1r and S here in these colors are going to have the proteolytic activity. And so here is where the numbering gets a little bit confusing. C1 recruits a molecule called C4. Now, all the numbers go in sequence except for C4, so you do have to remember this one. But so C1 recruits C4. It's a proteolytic activity. It's a protease reaction, so C4 is going to be cleaved. And when it's cleaved, it forms two subunits, C4a, which is the smaller subunit. C4a floats away, while C4b, which is the bigger subunit, attaches to the surface of the cell. And this process by which complement proteins attach to the surfaces of cells is fixation. So a lot of times we hear the term the fixation of complement. So when we say that, we mean the binding of complement proteins to the surface of cells. Okay, so now we've got C1 attached, we've got C4b attached, and we're going to see basically the same thing happen over and over now. One molecule attaches, it cleaves the next molecule in the pathway, and one of them floats away and one of them attaches. So I said they all go in order except for C4. So if we have C1 and C4, the next one we should expect to see is C2, now that we're going back in order. So C4b has proteolytic activity, which will cleave C2. Now, here's another source of confusion. In this case, the smaller subunit is C2b, and it floats away, and the larger subunit is C2a, and it attaches to the cell. So this is another point where you'll just have to memorize. But ultimately, C4b and C2a themselves form a enzymatic complex that we call the C3 convertase. So I said that all of the complement pathways converge on the formation of a C3 convertase. The C4b C2a complex is the C3 convertase of the classical complement pathway. So what does a C3 convertase do? Maybe by now you can predict that C4b C2a, this complex, is going to recruit a molecule called C3. So we're going back in order now. Remember, after C2 comes C3. And cleavage of C3 forms two products just like before. C3a, which floats away, and C3b, which can do a number of things. So C3b can also float away, where it can attach to the surface of pathogens, or it can stick around and attach to the surface of the original cell. And when it attaches to the original cell, it can complex with C4b and C2a to form a new enzyme called the C5 convertase. So now we've got to skip 4 because we already saw it. We'll go from C3 to C5. Okay, so you can probably see where this is going. A C5 convertase also cleaves C5. But I want to stop here because we just want to talk about how each pathway gets started up to the point where they make a C3 convertase. So our C3 convertase is here. It's converting C3. It's proteolytically cleaving it into either C3b or C3a. What I want to point out here is that these cleavage products of C3 are what initiate all of the different effector functions. So C3a is a very potent inflammatory signaling molecule. It can promote inflammation. When C3b coats the surfaces of pathogens, it serves as an opsonin, which promotes the process of phagocytosis. Or if C3b sticks around and forms the C5 convertase, the C5 convertase is actually the enzyme which initiates the formation of the membrane attack complex, and we'll talk about that in more detail later. So the formation of a C3 convertase is the pivotal enzymatic step in the complement cascade because the cleavage products of C3 are what really go on to promote all of the different pathogen control mechanisms of complement. So this is the classical pathway. We start with C1, which recruits all the various Cs after it, cleaving them in sequence, and ultimately resulting in cleavage of C3. And we're going to go further in this process to talk about the C5 convertase later. But this is the classical pathway. But there are different ways that we can get to a C3 convertase. So the classical pathway starts with C1 binding. As we'll talk about in the next lecture, complement can also get started through what we call the lectin pathway or the alternative pathway. So I'm going to talk about the lectin pathway and the alternative pathway in the next video, and we'll summarize all three of these pathways at the end of that video. Thanks.